Welcome everyone to this live podcast on Twitch where you can get the news from the research industry that how our researchers keep on researching on various domains and topics to keep our lifestyle more better and more sustainable. So, and yes, these articles are posted on our website named postscientist.com. If you are willing to check it out, please check it out. Otherwise, listening to this podcast will be enough for you guys because it acts like an archive on the daily basis, providing in the news from the research industry. So, let's get started without wasting further any time. The first topic of the day is scientists peel back ancient layers of banana DNA to reveal mystery ancestors. Bananas are thought to have been first domesticated by people 7,000 years ago on the islands of New Guinea, but the domestication history of banana is complicated. While their classification is hotly debated, as boundary between species and species subspecies are often unclear. Now, a study in Frontiers in Plant Science shows that this history is even more complex than previously thought. Okay. The results confirm that the genome of today's domesticated varieties contains traces of three extra as yet unknown ancestors. Here we show that most of today deployed cultivated banana that descend from the wild banana M. acuminata hybrids between different subspecies, at least three extra wild mystery ancestors must have contributed to this mixed genome thousands of years ago but haven't been identified yet, said Dr. Julie Sardos, a scientist at the Alliance of Biodiversity International and CIAT in Montpellier, France, and the study first author. Complex domestication history. Domesticated banana, except for the bananas in the Pacific, are thought to be descended from a cluster of four ancestors, either subspecies of the wild banana, Musa acuminata, or distinct but closely related species, M. acuminata, seems to have evolved in the northern borderlands between India and Myanmar and to have existed across Australasia approximately 10 million years ago before it was first domesticated. Oh, okay. A further complication is that domesticated varieties can have two diploid, three triploid, or four tetraploid copies of every chromosomes, and that many are also descended from the wild species M. balbicinia. Recent smaller scale studies suggested that even this already complex scenario might not be the whole story and that further ancestors related to M. acunata could have been involved in the domestication. The new result not only confirmed that this is indeed the case, they also show for the first time that these genes pools are common in domesticated banana genomes. Banana collecting missions. The author sequenced the DNA in 226 extract leaf extracts from the world's largest collection of banana samples at the Alliance of Biodiversity International and CIAT Musa Germplasm Transit Center in Belgium. Among these samples, 68 belonged to 9 wild subspecies of M. acunata, 154 to diploid domesticated variety descended from M. acuminata, and 4 more distantly related wild species and hybrid as comparisons. Many had previously been gathered in dedicated banana collecting missions to Indonesia, the islands of New Geneva, and the autonomous region of Bougainville. The researcher first measured the level of relatedness between cultivars and wild bananas and made family trees based on the diversity at 39,300 39,031 single nucleotide polymorphisms SNPs. SNPs. These use a subset of these even spread across the genome, with each pair demarcating a block of approximately 100,000 DNA letters. To statistically analyze the ancestor of each block, for the first time they detected traces of three further ancestors in the genome of all domesticated samples for which no matches are yet known from the wild. Mystery ancestors might survive somewhere. The mystery ancestor might be long since extinct, but our personal conviction is that they are still living somewhere in the wild, either purely described by science or not described at all, in which case they are probably threatened, said Sardos. Sardos et al. Have a good idea where to look for them. Our genetic comparison shows that the first of these mystery ancestors must have come from the region between the Gulf of Thailand and the west of the South China Sea, the second from the region between the North Borneo and the Philippines, the third from the island of New Geneva, could help breed better bananas. Which useful trait these mystery ancestors might have contributed to domesticated banana is not yet known. For example, the crucial trait of padenthrocarpy, fruit setting without the need for pollination, is thought to have been inherited from acuminata, while cooking bananas own a large chunk of their DNA to the species or perhaps separate species M. acuminata Bansky. Second corresponding author Dr. Matthew Rod, likewise at Biodiversity International, said identifying the ancestor of cultivated banana is important as it will help us understand the process and the path that shaped the banana diversity observed today, a crucial step to breed bananas of the future. Breeders need to understand the genetic makeup of today's domesticated diploid bananas for their crosses between cultivars, and this study is a major first step toward the characterization in great detail of many of the cultivars. Sato said, based on the result, we will work with partners to explore and genotype wild banana diversity in the three geographic regions that are study pinpointed with the hope to identify these identified unidentified contributors to cultivated bananas. It will also be important to investigate the different advantages and traits that each of the contributor provided to cultivated bananas. Hmm. I mean, interesting finding, interesting results. So, nothing to say more about it. We are moving towards next topic. The thinking undead. How dormant bacteria calculate their return to life. Okay, okay, let's see. Facing starvation and stress conditions, some bacteria enter a dormant state in which life processes stop. Shutting down into deep dormancy allows the cells called spores to withstand punishing extreme of heat, pressure, and even the harsh condition of outer space. Eventually, when conditions become favorable, spores that may have been dormant for years can wake up in minutes and spring back to life. Spores wake up by rehydrating and restarting their metabolism and Psychology, but until now, scientists did not know whether spores can monitor their environment in their sleep without waking up. In particular, it was not known how spores deal with vague environmental signals that do not indicate clearly favorable conditions. Would spores just ignore such mixed conditions or take note? University of California, San Diego biologists have solved this mystery in a new study published in the journal Science. Researchers in the School of Biological Science discovered that spores have an extraordinary ability to evaluate their surrounding environment while remaining in a psychologically dead state. They found that spores use stored electrochemical energy acting like a capacitor to determine whether conditions are suitable for return to normal functioning life. Okay, this work changes the work we think about sports, which were considered to be inert objects, said Girolo Seol, a professor in the Department of Molecular Biology. We show that cells in a deeply dormant state have the ability to process information. We discovered that spores can release the stored electrochemical potential energy to perform a computation about their environment without the need of metabolic activity. Many bacterial species form spores, partially dehydrated cells surrounded by resilient protective coat as a survival strategy that allows them to remain dormant for the thousands of years. Such a remarkable capability makes them a threat in the form of bacterial anthrax as well as a contamination hazard in medicine and the food industry. Soul and his colleagues tested whether dormant Bachelor's subtitles spores could sense a short-lived environmental signals that were not strong enough to trigger a return to life. They found that spores were able to count such small inputs, and if the sum reached a certain threshold, they would decide to exit the dormant state and resume biological activity. Developing a mathematical model to help explain the process, the researcher discovered that spores use a mechanism known as integrated and fire based on fluxes of potassium ions for appraising the surrounding environment. They found that spores responded to even short lived favorable signals that were not enough to trigger or exit from dormancy. Instead of waking up, spores release some of their stored potassium in response to each small input and summed consecutive favorable signals to determine if conditions were suitable for existing. Sorry, exiting. Such a cumulative signal processing strategy can reveal whether external conditions are indeed favorable and prevent spores from jumping the gun into a world of unfavorable condition. Hmm. The way spores process information is similar to how neurons operate in our brain, said Sewell. In both bacteria and neurons, small and short inputs are added up over time and to determine if a threshold is reached. Upon reaching the threshold, spores initiate their return to life, while neurons fire an action potential to communicate with other neurons. Interestingly, spores can perform this signal integration without requiring any metabolic energy, while neurons are among the most energy dependent cells in our bodies. The researcher believe the new information about spores reframes popular ideas about cells in extremely dormant states that seem dead. Such findings hold implications for evaluating life on objects such as meteors as well as space missions seeking evidence of life. Oh, okay. This work suggests alternate ways to cope with the potential threat posed by pathogenic spores and its implication for what to expect from extraterrestrial life, said Seoul, who holds affiliation with the San Diego Center for System Biology, Biocircuits Institute, and Center for Innovation. If scientists find life on Mars or Venus, it is likely to be dormant state, and now we now know that a life form that appears to be completely inert may still be capable of thinking about its next steps. Authors of the science paper include Kyoto Kishu, Lincia Guillermo Lepardo, Colin Weatherwax, Jamie Lamb, Ian Chelmune, Emmanuel Theodorkis, Jordi Garcia Oljelovo, and Guriel MCO. So, I mean, this scientist paper and researchers and amazing finding give the amazing finding about how an extraterrestrial body or bacteria which is being dispersed or extinct from the life can evaluating those things and uh, use their use other organisms to survive for on their self. So I mean, okay, it's an interesting finding because it's like he said in the article itself, it's going to be see in the other planets too. So yep, these are the findings given by this scientist. So yep, interesting. Moving on towards next topic: shrine discovered in Egyptian temple with evidence of previously unknown rituals. It's going to be an archaeological article, so let's see. The Sikyath Project Research Team, directed by Professor John Allard Grossman from the Department of Antiquity and the Middle Age Studies at the UAB, recently published in the American Journal of Archaeology. The results obtained from the January 2019 excavation season at the ancient seaport of Berenki, located in Egypt's eastern desert. The paper described the archaeological dig of a religious complex from the late Roman period, 4th to 6th century CE, named the Falcon Shrine by researcher and located within the northern complex, one of the most important buildings of the city of Berenki at that time. The site, which was excavated by the Polish Center of Mediterranean Archaeology and the University of Delaware, was a Red Sea harbor founded by Ptolemy II Philadelphus 3rd century BC and continued to operate into Roman and Byzantine periods when it was returned into the main point of entrance for commerce coming from Cape Horn, Iberia, and India. Okay. Within this chronological period, one of the phases yielding the most new discovery was the one corresponding to the late Roman period from the 4th to 6th century CE, a period in which the study the city seemed to be partially occupied and controlled by the Belmes, a nomadic group of people from the Nubian region who at the moment were expanding their domains throughout the greater part of Egypt's eastern deserts. In this sense, the northern complex is fundamental in providing clear evidence of a link with the Belmes people, thanks to the discovery of inscriptions to some of their kings or the aforementioned Falcon Shrine. Researchers were able to identify a small traditional Egyptian temple, which after the 4th century was adapted by the Belmes to their own belief system. The material findings are particularly remarkable and include offerings such as harpoons, cube-shaped statues, and a stele with indications related to religious activities, which was chosen for the cover of the journal Current Issues, says UAB researcher John Allard. The most remarkable consecrated element found was the arrangement of up to 15 falcons within the shrine. Most of these headless, although burials of falcons for religious purposes had already been observed in the Nile Valley, as had the worshipping of individual birds of the species. This is the first time researchers discovered falcon buried within a temple and accompanied by eggs, something completely unprecedented. In other sites, researchers had found mummified headless falcons, but always only individual specimens, never in group, as in the case of Berenki. The stele contains a curious inscription reading, "It is improper to boil a head in here," which, far from being dedication or sign of gratitude as normally correspond to an inscription, is a message forbidding all those who enter from boiling the heads of the animal inside the temple, considered to be profane activity. John Allard says that all these elements point to intense ritual activities combining Egyptian tradition with contribution from the Belmes, sustained it by the theological base, possibly related to the worshipping of the god Konzu. He goes on to say that the discovery expand our knowledge of these semi-nomad people and the Belmes living in the eastern desert during the decline of the Roman Empire. Interesting. Very interesting finding. Okay, moving on towards next topic. We are going to talk about the AI now. So let's see. Using AI to create more efficient math algorithms. Okay, let's see. A team of researchers at Google's DeepMind London has found that AI can find faster algorithms to solve matrix multiplication problems in the paper published in the journal Nature. The group described using reinforcement learning to improve math-based algorithm. A research briefing has also been published in the same journal issue, summarizing the work by the team in London. Math came up quite often in computer programming, generally as means for describing and then manipulating representation of real world phenomena. For example, it is used to represent pixels on a computer screen, weather conditions, or nodes in an artificial network. One of the major ways that math is used in such instances is to perform calculation on matrices. When programming games, for example, matrix describe possible movement options. For such movements to be realized, matrix are often multiplied or added together. Sometimes both actions are needed. This requires a lot of work, especially as the matrices grow larger, which is why computer scientists have spent a lot of time and energy developing more and more efficient algorithm to get the job done. In 1969, for example, mathematician Walker stretched and figured out a way to multiply 2 into 2 matrix using just 7 multiplication operation rather than the 8 that had been the standard. In this new effort, the researcher had a deep mind wondered if it might be possible to use reinforcement learning based AI system to create new algorithm with fewer steps than those now in use. To find out, they looked to gaming system for inspiration. Nothing that most are based on reinforcement learning. After building some preliminary system, the team turned its focus to tree searching, which is also used in programming. It is means for a system to look at various scenarios given a particular situation. When applied to multiplying matrices, the researcher found that the converting an AI system to give to a game allowed for searching for the most efficient way to get to a desired outcome, a mathematical result. The researcher tested the system by allowing it to search for review and then use existing algorithm using reward as an antecedent to pick that one that was most efficient. The system learned about the factor that contribute to matrix multiplication efficiency. Next, the researcher allowed the system to create its own algorithm, again looking to improve efficiency. The researcher found that in many instances, the algorithm chosen by the system were better than those that had been created by their human predecessor. Predecessors. Okay. Moving on towards next topic. Sample match, a model that automatically retrieves matching drum samples for musical tracks. Hmm. Machine learning based computational models have been successfully applied to a broad range of complex information processing tasks, including those that involve retrieving specific data items from large archives. Researchers at the Sony Computer Science Laboratory (CCL) in France have been trying to develop machine learning techniques that could help music producers to easily identify and retrieve specific audio samples from a database. Hmm. To this end, Stephen Lathner, a researcher at Sony CSL, recently introduced Sample Match, a machine learning based model that can automatically retrieve drum samples that match a specific music track from large archives. His model is said to be presented in December at the ISMIR 2022 conference, a leading event that focuses on music information retrieval. Our music team at Sony CCL is working on AI that could make the life of music producer easier. Stephen Latner, one of the researchers who carried out the study, told Tech Explore, in music production, there are many tasks of or which AI could be valuable. One such task that is currently relatively tedious in drum sample selection. Drum sample selection is the process through which a music producer must search for drum samples that would work well with specific drums less music track. As drum sample libraries are typically large, identifying suitable drum samples can be very time and energy consuming. Currently, music producers only have access to a few rudimentary computational tools designed to exist them with drum sample selection processes. These primarily include filtering a large dataset by tags or keywords. Hmm. Okay. A few years ago, Leitner set out to develop a new system that could retrieve drum samples in more initiative and effective way due to limitation of technology available at the time. However, this system needed to be relatively complex. I found that the system I previously created was not a very elegant, so I didn't publish it. Leitner explained. Okay, with the recent advance in constructive learning and improvement in net neural network encoders, it has become much easier to estimate if two data points fit together. As a result, the system became more general and my method could be used to estimate the fit of many kinds of sounds. Okay. When using sample math, musicians can put their track into the system at any stage of production. The system then automatically sort a drum sample library based on what is calculated would match best with it. Oh, interesting. Leitner trained sample match use a large dataset of 4,830 electronic music tracks and 885 famous pop rock tracks. Specifically, he used audio pairs of instrumental music, that is synthetic basses, bass, guitar, pad, strings, chord, keyboard, and vocals, and matching drum tracks. Sample match was trained an audio pair that we knew would match, Ladner said. Now, when we show a new pair to the model, it will provide a matching score. While there are already systems that match audio samples using extracted musical features, their retrieval quality depends on the predefined features and the type of samples. From done samples, it is not even clear which features we should look at to compute a matching score. While Leitner trained his model to learn what drum samples match a specific track, it could also be used for other forms of audio matching by using different training sample pairs. In fact, sample match could also be taught to retrieve matching bass, guitar, or other instrumental tracks. Hmm. Some aesthetic choice a musician perform in music production are still mysterious. Ladner said. While it is obvious that an instrument should not be played out of key with drum samples, there is no theory why some fit your track and some don't. By showing samples, a computer can now learn the aesthetic principle we apply when listening. In some way, the computer learns to listen like a human. In the future, the audio retrieval model created by Latner and his colleagues at Sony CCL could assist music producers in sourcing suitable drum samples or other instrumental sample for their tracks. In addition, a close analysis of how the system learned to organize data could help to devise new theories that might guide music production efforts. More specifically, the reverse engineering of the system might allow the researcher to outline some general rules that musicians should follow when mixing their music. In our future works, we want to combine this method with our drum GAN technology to generate drum samples that match a given track directly, Leitner added. Meanwhile, we also want to extend sample match to other kinds of samples. Right, okay. I mean, it's an amazing model which is being used by the many of the musicians and music producers. So great, great. I want this. I mean, I want this model in the market itself by any of the company. But do please do other. It's going to produce more music and more efficient music, which is more relaxing in nature and basically providing you the waves that your brain can actually function more efficiently. So we need these kind of models, not just the music. We need this kind of music which needs to relax your mind, relax your brain, and of course, if you are doing um, if you are into physics, then you know that how music can affect your brain and neuroscience things. So yep, moving on towards next topic: creating a mouse embryo from stem cells to learn more about the mammalian development process. Okay. A team of researchers at the California Institute of Technology, working with one colleague from the Francis Crick Institute and another from the University of Cambridge, both in the UK, has developed a way to grow mouse embryos without use mouse eggs or sperm to learn more about early mammalian development. In the paper published in the journal Nature Cells Biology, the group described using several types of stem cells to grow mouse embryos. Okay. Prior research has shown that mammalian embryos differentiate into different types of cell masses as they develop. Researchers have also found that the stem cells are involved in the process, but the mechanisms responsible are still unknown. In this new effort, the researcher used three different kinds of stem cells to grow a mouse embryo that matured to the point of having a beating heart and the beginnings of a brain. To create such embryos, the researcher first studied communication between stem cell group in naturally developing mouse embryos. They learned to recognize these elements that went into such communication and the means by which it was carried out. In essence, they deciphered the code. They then isolated three main types of stem cells that made up the cell that made up the cell masses in early embryo development. Pluripotent, which eventually grew to become body tissue, and two other types that grew to become the embryonic sac and placenta. They also noted the quantities of each type of stem cells. The next type was to attempt to create a mouse embryo from scratch using the three types of stem cells in a lab setting. With careful tending, the researcher grew an embryo that matured enough to allow for study of its development. To test further, the researcher repeated the procedure but added genetically engineered cell to see how it impacted maturation of the embryo. They found that they could replicate some of the same brain development issues that has have been seen in human embryos. They suggest their work could also help explain what goes wrong when mice or people miscarry. Oh, okay. So, I mean, it's basically providing the information that how mammalian development process and how organism or embryos differentiate with the parent cells and into the another body. So, I mean, basically discovering that how a new birth can actually be done in the mammalian development process. So, yep, of course. And of course we use mice because we think mice is are similar to the mammalian, so it's easy for to test on them. So yeah, moving on towards next topic: new form of silicon could revolutionize semiconductor industry. Oh, okay, let's see. After a 10-year research study that started by accident and was met with skepticism, a team of Northeastern University mechanical engineer was able to synthesize highly dense ultra narrow silicon nanowires that could revolutionize the semiconductor industry. The research appears in Nature Communication. Jung Jun Jung, okay, Northeastern professor of mechanical and industrial engineering, say it might have been his favorite research project. Everything is new and it's required a lot of perseverance, says Jung, who specialize in engineering and application of nanostructure system and previously studied carbon nanotubes. Jung and his collaborator, including another Northeastern professor of mechanical engineering, Munish Upmanyu, have achieved a major advancement in nanowire synthesis by discovering new highly dense form of silicon and mastering a new scalable catalyst free etching process to produce ultra small silicon nanowires of 2 2 to 5 nanometers in diameter. About 10 years ago, students brought Jung attention to an unusual results of an experiment they were conducting using silicon wafers. The material he saw under an electron microscope was different from the one they intended to produce, Jung says. He decided to find out more about this substance and discovered that it was silicon with a very tiny wire like nanostructure. Jung says they were able to reproduce the new material, he says, but when they tried to improve the synthesis process, the nanowires didn't grow. The scientist and his team had to rewind and study from the beginning the synthesis mechanism and the material atomic scale, structure, and properties. Jung, an experimentalist, decided to enlist Upmanyu, who used theory, computer modeling, and simulation to understand material and explain experiments. I always need help from Munish to understand what is happening, Jung says. The scientist thought that maybe the substance resulting from silicon wafers during synthesis was not silicon at all. The material had a highly compressed structure, reduced by 10% to 20% compared to regular silicon, which normally is not stable in such a compressed state, Upmanyu says. Some of the colleagues and research reviews were of the same opinion. They would say this shouldn't be silicon or this shouldn't occur with silicon. Jung says, through the computational analysis and modeling, Upmanyu was able to show that despite unusual properties, the new material was formed of silicon with a very tiny layer of oxide on top, which probably helps sustain the compression, he says. This material is very promising, he says. That compression, I feel, is at the heart of all the interesting properties you see. One of the reasons silicon is widely used as semiconductor in microelectronics such as computer chips, integrated circuit, transistors, silicon diodes, and liquid crystal display (LCD) is that it is cheap and abductant upon uses. Of course, according to the Royal Society of Chemistry, it is the second most abductant element in the Earth crust after oxygen. Oh, but it does not occur uh, in its pure uncombined state in nature. Okay, it can found in the sand, quartz, flint, granite, mica, and clay, among other stones and many minerals. Oh, okay. In the 1970s, the thriving silicon computer chip industry even gave a new name to the southern region of the San Francisco Bay, Silicon Valley, which was popularized by Don Ho Efflord, an electronic news magazine reporter. However, traditional silicon cannot withstand high temperature and hence is limited to lower power application. It has a band gap of 1.11 electron volts. Band gap determines the energy needed to make electrons in the semiconducting material conduct electricity upon being simulated by external sources. The new material has an external wide band gap of 4.16 eV, a world record, Jung says. The ultra wide band gap implies that the material needs larger stimuli to conduct electricity but can operate at higher power, high temperature, and high frequencies. Silicon nanowires produced from this new material will be suitable for powered electronic transistor, diodes, and LED devices, Jung says. Hmm. Unlike regular silicon, the new material is highly resistant to oxidation. It is also photoluminescent, able to emit blue and purple light, which can be used for ultraviolet lighting and in blue light diodes. Jung and his research team also created a new method of producing silicon nanowires called chemical vapor etching, which removes material instead of glowing crystal. As a result, they can make nanowires that are 10 to 20 times smaller than the silicon nanowires currently used commercially. Previously known nanowire synthesis process use catalyst particles to grow silicon crystals. The crystal free aspect cannot be overstated enough as it eliminates the need to remove the catalyst after synthesis, which inevitably degrades the functional properties of the nanowires, Jung says. Sometimes catalyst particles become a part of the nanowire surface. He says their removal is almost impossible. At this point, the scientist can reproduce nanowires with control length of up to 100 micron. I feel a broad impacting going forward upon uses. This chemical vapor etching method that He Jung has pioneered is going to be useful for a host of other materials. You can think of not just electronic application, but any application where you want to have a small size dimension of a material made. It is very powerful. He says that new silicon material should be very attractive for the semiconductor industry. It can be used in military radios, radars, and photovoltaics like solar cells. Regular silicon bandgap does not allow to process ultraviolet light and use it for generating electricity upon usage. So, if you have a wide bandgap material which is cheap, abductant like silicon, now you can have very high efficiency solar cell. He says. Oh, I mean, oh, okay. It can be even used for harvesting solar energy underwater. Water absorbs the red infrared spectrum when you say so. Solar cell that can harvest blue and ultraviolet light become crucial. The new silicon nanowires can improve lithium-ion battery junctions, further adding some select material like phosphorus or nitrogen. A technique called doping can lead to other interesting properties and allow other application upon uses. Of course, he believes that various interesting quantum phenomena can be manipulated in these silicon nanowires due to their very small size, which makes this material promising for quantum information processing and maybe even quantum computing upon uses. Hmm. Several other engineering institutions and the globe contributed to this research, including Korea Institute of Science and Technology, Korea Advanced Institute of Science and Technology, Tokyo University of Science, University of Science and Technology of China, and Ruslan Polytechnic Institute in New York. The researcher, the research is not over. Of course, the scientists are still interested in understanding better all the chemistry behind the process and figuring out why the compression of this form of silicon is so stable. They want to optimize the etching process to produce a smoother surface and further scale it up for industrial application. You want to be able to understand the process so that you can manipulate it to what you want to do. Open you says they will be also looking for collaborators interested in making devices with this new silicon material. You want a new form of something you made to be adopted as widely as possible. I think commercialization and device integration is the key here. Upman says. Okay. So I mean, it's interesting that our researchers and scientists keep on finding these things of the silicon industry and converting other source of energy or whatever, uh, which will provide more energy to the computers or any other material available in the market. So yeah, or available in our environment, of course, not getting harm or sustainable process with those things. So yep, I mean, great finding, great. So yep, moving on towards next topic. Researcher developed thermoformable ceramics, a new frontier in materials. Okay, it was one of those happy accidents of science. Northeastern Professor Randall Obb and PhD student Jason Bice were working on a product of 40 university client and would up with an entirely new class of material. The discovery of an all ceramic and that can be compression molded into complex parts, an industry breakthrough, could transform the design and construction of heat emitting electronics, including cell phones and other radio components. Our research group lies are very much situated at the bleeding edge of technology, says Erb, an assistant professor of mechanical and industrial engineering who leads the DEPS lab at Northeastern. Okay, things break a lot, and every once in a while, one of those breaks turn out to be good fortune. Last July, Erb was in his northeastern lab with boys who has since earned a mechanical engineering PhD. They were testing an experimental ceramic compound as a part of hypersonic project for the industrial partner when something appeared to be wrong. We blasted it with blowtorch, and while we were loading it, it unexpectedly expectedly deformed and fell out of the fixture. Herb says. We looked at the sample on the floor thinking that it was a failure. Closer examination gave way to a revolution. We realized it was perfectly intact. Herb says it was just shaped differently. Ceramic tend to fracture or even explode from thermal shock when subjected to extreme heat changes and mechanical loading, but their sample had deformed gracefully. We tried it a few more times and realized that we could control the deformation of says, and then we started compression modeling the material and found that it was a very fast process. Its underlying microstructure uniquely allows the oil ceramic to quickly transmit heat and flow effectively during the molding process. The ceramic can be formed into executive geometrics and exhibit impressive mechanical strength and thermal conductivity at room temperature, says Herb, whose finding were recently published in Advanced Materials. Herb and Bice are developing the product via their startup Fourier LLC, named from after the French mathematician Joseph Fourier, who studied the heat flow in ceramics 2 centuries ago. Fourier has received $50,000 Spark Fund award from Northeastern Center of Research Innovation. Oh, okay. It's unique, of course, it's unique theoreformable ceramics from what we have seen and read don't really exist, by says. So it's a new frontier in materials. Of course, the new product has the potential to introduce two industry improvements, starting with its efficiency as a heat conductor that can cool high density electronics. Yes. In general, cell phone and other electronics are fitted with a bulky layer of aluminium which is necessary to draw heat away from the unit. Yes. Our material can be less than a millimeter thick, which presents a low profile solution by says. Of course, it can be molded to conform to the surface that you are trying to cool. The photonic crystal based ceramic allows heat to flow without electron transport, says Herb. It doesn't interfere with radio frequency RF of cell phones and other systems. If you put an aluminum heatsink into a RF component, you're basically introduce a series of antenna to interact with RF signals. Herb says instead, we can put our boron nitride based material in and around an RF component, and it is essentially invisible to the RF signal. The other improvements is that it can form fit directly to the electric component. Eco ST Germain, a fifth year mechanical engineering student at Northeastern who is serving as a ceramic research and R&D engineer at 4 year, demonstrated the ceramic non-Newtonian behavior by subjecting a clumpy slurry of it to vibration that is normal to the manufacturing process. It instantly liquefied and organized the structure of the material. Such slurries are used to produce the moldable ceramics. So it's going to be the next alternative of the silicon. I mean, basically he's providing those kind of things. So yeah, I mean, let's see. Herb and Bice believe that they will able to form fit and all ceramic material to all kinds of electronic components. The ceramic will be thinner, of course, lighter and more efficient than the metals currently in use. Hmm. Bice has helped launch the startup from Munich, okay, where his wife has started a new job. Oh, okay. Launching a company that is Boston based while I'm in Germany adds some interesting complication to things, but also opportunities, says Bice, who with Herb is engaging in the customer discovering in both Europe and the US. I mean, he deserves it. He deserves it. If it's going to be the next silicon alternative, so I mean, great, great, great. I mean, great, great research. Uh, I, I'm more um passionate about this thing. I mean, yes. I hope this startup will be launched more in the available in every market and going to be disrupt everything. So yep, I mean, great, great, great finding and great research, of course. And we are moving towards the startup field. So yeah, oh, of course. Okay, moving on to our next topic. Light-based therapy weakens antibiotic-resistant bacteria. Okay, okay, let's see. Antibiotics are standard treatment for fighting dangerous bacterial infections, yet the number of bacteria developing a resistance to antibiotics is increasing. Researchers from Texas A&M University and the University of Sao Paulo are overcoming this resistance with light. Okay, the researcher tailored antimicrobial photodynamic therapy, a PDT, a chemical reaction triggered by visible light, for use antibiotic resistant bacteria strains. Results showed that treatment weakened bacteria to where low doses of current antibiotics could effectively eliminate them. Using a PDT in combination with antibiotics creates a synergy of interaction working together for the solution, said Wordsworth, Yulsworth, Yakulsworth. Sorry uh, if my pronunciation of this name is wrong, so please forgive me. University professor in the Department of Biomedical Engineering at Texas A&M and co-director of the project. It's a step in the right direction against resistant bacteria. Hmm. The research result was published in the proceeding of the National Academy of Science (PNAS). Ultraviolet light was first used to sterilize bacteria over 100 years ago. The treatment was based on the work of Niels Finson, who won the Nobel Prize in Psychology in 1903 for using filtered sunlight, the higher frequency or ultraviolet spectrum, as a cure of for skin tuberculosis. Phototherapy advances faded in popularity a few decades later when antibiotics became the weapon of choice against bacteria. Antibiotic resistant bacteria showed up soon after antibiotics were first used. If antibiotic treatment stops before bacteria are fully killed, the remaining bacteria develop a resistant or immunity to the antibiotic. That immunity transferred to every new bacterial cell. So more potent antibiotics or new treatment methods are needed to overcome the growing resistance. Photodynamic therapy was a forgotten tool. Yakult said, yet bacteria cannot overcome it. There is some resistance. Some human cancer cell therapies already use a PDT to prevent the growth of abnormal cells, but treating resistant bacteria with it is still a novel approach. The researcher began their work by choosing the bacteria and the three main parts of a PDT needed to combat it: molecular oxygen, light, and photosynthesis. Uh, Sensitizer, something that creates a reaction between oxygen and light. An already FDA approved dye called methylene blue served as a photosensitizers. The light source were specially constructed panels of 25 LEDs in reflective cones built by the Technical Support Laboratory by the Sao Carlos Institute of Physics. Methicillin resistant Staphylococcus aureus served as the bacteria, and the researcher grew culture and their blue dye in them to ensure photosynthesizers alone would not affect the bacteria. Most of the lab occurred in the Texas A&M Health Science Center under Paul Day Figueredo, professor in the Department of Microbial Pathogenesis and Immunology in the College of Medicine. At first, the team used a PDT by itself, various light strands, duration, and in a specific series of follow-up treatments, and to log the bacterial response. The idea was to find the lowest dose and the shortest series that could weaken the bacterial membranes and other resistant mechanisms. Cell recoveries and reproduction revealed how many generations it took before antibiotic resistance returned. Next, the researcher added measured levels of and combination of antibiotics at a different time interval after a PDD treatment to note the weakened bacterial response. The use of antibiotic with a PDD is a unique idea, Yikelvo said. We can use lower doses of both to achieve our goal, in contrast to using one of the other at higher doses that could have side effects. The goal is to shorten the treatment time and reduce the dosage to the lowest levels needed. Getting medical care down to one doctor visit is especially important to Vandalia Bagnotto, professor in the Department of Physics and Material Science at Sao Paulo and co-director of the project. He's trying to improve recovery odds for population in remote areas of Brazil where patients might only see a doctor once per illness without any chance for follow-up care. The US Department of Defense is following the project closely because battlefield wood infection also happen in remote locations and must be dealt with quickly. So far, the results are positive. The resistant bacteria weakened by PDT treatments were killed with far lower doses of current antibiotic as a benefit. These therapies reduce the need for battling resistant bacteria with more potent and expensive antibiotics that take years to produce. Future work for the project will involve more timing and doses investigations and tests on other resistant bacteria strains to see if this effectiveness is universal. Imagine the real life application. Yakalvo said, you visit a doctor who uses an ointment and shines a light on the infected, and then you're done. It it would be a quick and harmless treatment as needed. Okay, okay, okay. I mean, it's similar like a Li-Fi system that providing. I mean, Li-Fi basically provides you the internet or through the light, but I mean, it's like a similar thing, and that just you need a ointment and a shine of light on the infected area, and then you're fixed. Your your therapy is you are done with your body, and you are again going to your work and any other field of your work. So I mean, great, great, great. So yeah, moving on towards next topic. How tar digrades survive freezing temperature. It's a animal who's mostly, I guess, available on the frozen areas on the mountain regions. So let's see. Tardigrades are excellent at adapting to harsh environmental conditions. Back in 2019, Rafael Skills, a professor at the Institute of Biomaterials and Biomolecular System at the University of Stuttgart, proved that any hydrobiotic dried tar, uh, tardigrades can survive undamaged for many years without absorbing water, whether the age faster or slower in a frozen state, or whether aging even becomes to a halt was previously unclear. But the mystery has now been solved. Frozen tardigrades do not age. Tardigrades, also called water bears, belong to the family of nematodes. Their gait is reminiscent of that of a bear, but that is the only similarity. The tardigrades, which are barely 1 millimeter in size, have managed to adapt perfectly to rapidly changing environmental conditions. over the course of evolution and can dry out in extreme heat and freeze in cold conditions. They don't die, they fall into deep sleep, explains Skills and a sleeping beauty hypothesis. For a cell organism, freezing or drying out cause different kinds of stress, but tardigrades can survive both heat and cold equally unscattered. They no longer show an obvious sign of life, and this raises the questions of what happens to the animal internal clock and whether they age in this resting state. For tardigrades, which wait many years in their habitat for the next train, Ralph Skills and his team answered the question of aging several years ago in a fairy tale for the Grimm brothers, and the princess falls into a deep sleep. When a prince kisses her 100 years later, she awakens and still looks as young and beautiful as before. It is the same with tardigrades in a dry state, and therefore, that this is called the sleeping beauty hypothesis, sleeping beauty model. Hmm. During inactive period, the internal clock stops and only resumes running once the organism is reactivated, explains Skill. So tardigrades, which usually only live for a few months without periods of rest, can live for many years or even decades. Okay. Until now, it was still unclear whether this also applies to frozen animals. Do they age faster or slower than the dried animals, or does aging also come to an halt? The aging process stops even when frozen. To explore the skill and his team conducted several experiments in which they froze a total of more than 500 tardigrades at minus 13 degrees Celsius, thawed them out again and counted them, fed them and froze them again. This was repeated until all the animals died. At the same time, control groups were kept at constant room temperature, excluding the time in frozen condition. The comparison with the control groups showed an almost identical lifetime. So even in ice, tardigrade stops their internal clocks like Sleeping Beauty. Concludes Skill and his colleagues published their findings and approach in the Journal of Zoology. Of course. Okay. Okay. Moving on to our next topic. More like a news and a topic of the SARS-CoV-2 virus. So, see, SARS-CoV-2 virus found to mimic a protein that packages DNA, preventing transcription. A team of researchers at the University of uh, Pennsylvania, working with a colleague from the University of Texas Medical Branch and another from Boston University and Boston Medical Center, has found that the SARS-CoV-2 virus produces a protein that mimics a protein that packages DNA, preventing transcription that would normally play a role in an immune response. In their paper published in Nature, the group described how they compared proteins that package human DNA with proteins produced by the SARS-CoV-2 virus and what doing so showed them. Lisa Thomnan and Volker Thiel with the Institute of Virology and Immunology, Bern and Mithil Hossain, have published a news and views a piece in the same journal issue describing the work done by the team on this new effort. As the pandemic has gone on and lessened, medical researchers have continued to study the virus behind it, and one of the areas they have focused on is its remarkable ability to suppress a response by host cells in those infected. In this new effort, the researchers have taken a closer look at the relationship between proteins that package DNA in human cell and protein produced by the SARS-CoV-2 virus. Prior research has shown the SARS-CoV-2 virus produces the three main types of proteins: those involved in replication, those involved in structural process, and a third that are known as accessory proteins. The third type is involved in a wide range of activity. Prior research has also shown that human DNA is packaged in protein known as histones. They serve to hold the DNA strands in place and to serve as gatekeepers for material that read the strand and use that information to engage in activities such as signaling to other body parts. Some such signaling is involved when a host is infected to urge defense mechanism to fight the intrusive agent. In this new effort, the researchers have found evidence that suggests that one of the accessory proteins produced by the SARS-CoV-2 virus, ORF8, is able to mimic at least one type of histone protein CAR2A that packages DNA, and by doing so dampens the type of signaling that is supposed to occur when a host is infected, reducing the immune response. I mean, I wanted to explain it, but I'm behind time, so moving on towards next topic. Using machine learning to find an optimal mixture of metals to create a desired alloy. Okay. A large team of researchers at the Max Planck Institute for Unserforksburg GmbH, working with colleagues from Technische Universität Darmstadt, Delft University of Technology, and Keth Royal Institute of Technology, has found that it is possible to use machine learning to help metallurgists find the optimal mixture of metals to create a desired alloy. In their paper published in the journal Science, the group described the three-step process and how well it worked when tested. Qiu Miu Hu and Ryu Yang with the Chinese Academy of Science Institute of Metal Research have published a perspective piece in the same journal issue outlining the work done by the team of this new effort. Humans have been mixing metals to suit their needs for thousands of years, and in so doing have learned a lot about creating alloys, but finding just the right mix has almost involved some degree of inspiration, patience, and chance. So much alloys have been created by starting with one main metal such as iron, adding small amounts of other metals to see what characteristics resulted. Over the past few decades, things have begun to change. However, some researchers have begun making alloys that have equal parts of several metals. Creating such alloy with desired feature is, of course, much more challenging. In this new effort, the researcher applied machine learning to help this with the processes. They began by reducing the text space to just one application, creating alloys that do not expand and contract very much when exposed to temperature changes. To create a machine learning application, the researcher looked for and found characteristics of metal that could be used for comparison purposes, and then taught the system using information in currently available databases. In so doing, they developed a process for finding an alloy that would suit a desired purpose. The process by the team was narrowed and down to three basic steps. First, they generate new mixtures using model based on information held in the database describing metal characteristics. Next, they use a second model to help predict the properties of certain alloys they created using the first step. The final step involved studying the alloys candidates produced by the system and choosing some to test in the real world. Using this system, the researcher derived 1,000 candidates, which were narrowed down to just three alloys. They then created the three alloys using the mix described by their system and tested their physical property. The team then trained the system on data derived from the real alloys and repeated the whole process in all. They ran it 7 times and found an alloy with a smaller thermal coefficient than the current record. So, yep, I mean, using of machine learning to the optimal senses. So, yep, interesting. Okay, as I previously discovered that we have previous podcast that we have started a newsletter for you guys, providing you the information that is not easily available on the surface web. So if you are into that kind of information, please subscribe to this newsletter. Otherwise, it going to be anything else. I mean, it's nothing. So if you're interested in that kind of information, subscribe to this. Otherwise, leave it. And till then, keep researching. Sorry. I also wanted to tell you that uh, we are uh, becoming a more member, so it's a requirement to subscribe to this newsletter, and of course needed money to have a sustainable team to provide you the information without having uh, any propaganda website. And yes, we are working on our website. We are most likely to be live by the next week, or yes, more likely live on the next week, and the every other thing will be provided and explained by you guys. So yep, in the end I just wanna say, you guys, keep researching, stay curious, stay healthy.